lipid capsule, which makes it easier for it to assimilate into the animal cell. This is what's called lipofaction. We did this using COS7 cells, which are cell lines derived from, as I mentioned before, a monkey, because that is very similar to human cells. And any observations we make here, we want to at least be similar to the effects that they would have in human cells. So we just transfect this using a turbofect kit, making so that the plasmids are inside these lipid capsules, and it's very easy to transfect into these animal cells. But once we do that transfection, we need to actually put these altered animal cells onto a microscope slide so that we can view it. But right now, it's attached to a glass slip in culture media. We need to move it so that we can properly observe these cells. And to do that, we do what's called trypsinization. We apply an enzyme called trypsin that breaks down the proteins on the outside of the animal cells, making it so that they are no longer attached to that glass slip. They become suspended in their culture medium, making it very easy to move them to another place, container, or in our case, microscope slide for imaging. But in order to actually see anything under the microscope, we need to do immunostaining. What is that? It's where we use antibodies to identify certain proteins in those cells. We use primary antibodies, which are antibodies from an organism other than the host organism for these cells that binds to this specific protein. Next, we use a second type of antibody with a fluorescent tag on it that searches for antibodies from the organism that the primary antibody is from. So for example, we could get this primary antibody from a mouse, and then a second antibody from a different organism that's searching for mouse antibodies. That way, we can make sure that these second antibodies will only find the primary antibodies. With those fluorescent tags, those proteins will then be visible under a microscope, and we can observe the structure of the microtubules to see the effects of the tau protein. Before we do that though, we need to do what's called fixation, where we put cells in PFA, which is paraformaldehyde, or methanol. Those aren't important. To stop the biochemical processes. We don't want the cells moving around while we're trying to image, or else the image could be blurry and our results could be unclear. From there, the cells can be mounted on a microscope slide and viewed. So here are our results. The red color is alpha-tubulin, which is the primary protein that makes up microtubules. Green is the GFP. Our plasmids had the gene for this protein, green fluorescent protein, so it's very obvious which cells took up the plasmid, because not every cell takes it in. The blue is the cell nuclei, using a stain called DAPI. This is to make sure that what we're looking at is actually cells, and as we can see, all of these are cells. So here we have a negative control. Nothing was added to these, except for the antibodies. These are normal cells. We can see the stringy microtubule structure in these. So here it's a little more clear. See, there's a lot of strings coming out throughout the cell structure. Here we have an A is the mutant type. In one of the edited cells, we can see that there are no strings. It looks just like a solid blob of red. Because in the mutant type, the tau is not able to bind to the microtubules. So the tau isn't able to stabilize them. As a result, they can't form these long structures, and they end up being very small, which is why it just looks like pure red. And B, our wild type, we would expect it to look very similar to the negative control, with just these long string structures. And we can see long string structures in the wild type cell, but it doesn't look the same. It looks a little off. Why is that? So in the negative control, we can see, it looks normal. The microtubules are already being properly stabilized. So if we add even more stabilization with the tau, the microtubules become overstabilized. They still form these long strings, but they aren't able to detach. So we can see that they're much longer than in the negative control, which tells us that the microtubules need to be very carefully maintained. We can't just add extra stabilization. So this shows us that tau has a very large impact on the stabilization of microtubules.
Adding on to what he